Hello everyone, welcome back to the next video in the Chapter 3 lecture series for human physiology. In the previous video, we looked at the structure and composition of the cell membrane. So now in this video, we're going to start talking about some of the things that you find in the cell membrane and how that influences what the cell membrane is capable of in terms of physiological processes. So the first thing to understand is that the cell membrane is not exclusively made out of phospholipids. So I like to think of it kind of like the ocean. The ocean is not exclusively made out of water. There's other things in the ocean besides water, right? There's salt, there's icebergs, there's all kinds of aquatic life. The cell membrane's the same way. Now, you can certainly think of the phospholipids as being like the water, but like water, there are icebergs floating in it, and that is what we would consider to be the membrane proteins in this. There are proteins that actually are embedded in the plasma membrane, and they float in the membrane like icebergs at sea. In fact, there are so many proteins in the plasma membrane that proteins are actually the majority component. That's kind of wild to think about. Now, proteins we learned all about in the previous chapter in terms of what proteins are made of and how different amino acids and different primary sequences and different folded structures give proteins different functions. But what we have not yet come to appreciate is that proteins do literally everything in physiological processes. No matter what kind of physiological process you want to talk about, proteins are how those things get done. So specifically, if you're talking about the cell membrane, which membrane proteins are in a given cell's plasma membrane, that's going to have a lot to say about what that cell is capable of. Membrane proteins give special functions to the cells that have them. Okay, now before we get too into what those functions might be, let's categorize these membrane proteins. The first are called integral membrane proteins. These are the types of membrane proteins that go all the way through from one side to the other. They span the entire width of the membrane, and these are usually going to include proteins that we are going to talk about a lot in this chapter: channel proteins, carrier proteins, junction proteins. And then receptor proteins are a type of integral membrane protein that is going to be exquisitely important once we start talking about cell-to-cell communication. And then you also have peripheral membrane proteins. These are proteins that are anchored to one side of the membrane or the other, either the extracellular fluid side, which is this side up here, or the intracellular fluid side, which is this side down here. So this would be a peripheral protein, this would be a peripheral protein, this would be a peripheral protein. The ones that you see going all the way through, those are integral membrane proteins, so it's important to know the difference. In addition to proteins, you can also find carbohydrate chains in the plasma membrane, but you only are going to find them in one specific place. You can find them attached to either proteins, in which case we call them glycoproteins, or you can find them attached to the phospholipids, in which case we call those glycolipids, but you only find them on the leaflet of the membrane that faces towards the extracellular fluid side. That's why these little chains of sugars that you see here are all facing towards the same direction, towards the extracellular fluid. You do not find any carbohydrate chains down here on the side of the membrane that faces inward towards the intracellular fluid. Make sure you remember that. You only find sugar chains on the outside part of the membrane. And what these sugar chains are there for is to essentially give your cells a fingerprint. So everyone has their own unique fingerprint. Everyone's cells are going to have their own unique carbohydrate fingerprint as well. And your immune system is very in tune with what your normal sugar chain fingerprint looks like, so that if a foreign invading cell like a bacterium or a bad blood transplant or something like that occurs, your immune system cells can recognize sugar chains that should not be there. And then if we look at receptor proteins, and we're going to talk a lot more in depth about receptor proteins in the next unit, but we'll go ahead and introduce them right now. Receptor proteins are a type of integral membrane protein that allows a cell to respond to a signal that originates elsewhere in the body. These signals come in all kinds of shapes and forms, hormones, neurotransmitters, and the like. We call any type of chemical signal that acts like this, in general, we call it a ligand. Ligands are small molecules that bind to specific parts on a receptor molecule, and what happens is once that ligand binds, this is going to lead to a change in that cell's function. So that basically what's happening here is that a cell elsewhere in the body releases this ligand molecule that travels either a short or a long distance to get to a target cell, the cell that is receiving the message. That ligand binds to a receptor on the target cell, and that cell responds in a specific way. We'll talk all about how hormones operate this way, neurotransmitters operate this way, so it's good to just go ahead and get an idea of what those discussions are going to look like. But if we want to put this into context for what we talked about in Chapter 1 with how homeostasis and negative feedback loops work, these ligands form the basis of your input and your output signals. These are the ways that cells in different parts of the body communicate with one another. Okay, so specifically for this chapter, we want to focus our attention on channel and carrier proteins, because these are the types of membrane proteins that act specifically in membrane transport, the movement of molecules from one side of the membrane to another. So these are integral membrane proteins that allow a very specific kind of molecule, or an ion, which is technically an atom and not a molecule, to pass through the membrane under a set of circumstances. Number one, that protein has to be open. If you look at this channel protein here, essentially what's happening here is it's kind of like a hollowed out, trying to think of the word, cylinder. It's like a hollowed out cylinder, so that if a molecule wants to pass through the membrane, it can just move in through what is essentially a tunnel that goes through the membrane. But these channels and these carriers can open and they can close. So if a molecule is going to be able to pass through, the channel has to be open, first of all. That is kind of common sense. And then number two, the cell has to express the protein to begin with. Express is just a fancy cell biology term that means the cell has to actually make that protein and stick it in the plasma membrane, and not every cell necessarily is going to do that. A good example are the kidneys. There are different cells in different parts of the kidneys that have different kinds of membrane transport proteins, different channel and carrier proteins, which is responsible for different parts of the kidneys filtering out salts and different ones filtering out water and things like that. So these are two things that you can't necessarily take for granted. You can't take for granted that every cell in the body is going to have glucose transporters. You can't take for granted that every cell in the body has sodium transporters that are always open. So these are things that we need to be thinking about. Okay, so we've got it in our mind that for a lot of cell transport processes, we're probably going to need those channel and carrier proteins, right? Well, what about the kinds of molecules or materials that need to be transported? What are some examples of those? Well, number one, nutrients like glucose, which is a carbohydrate, fatty acids, which are lipids, amino acids, oxygen, all the sorts of things that we get from our food or from our environment, they need to be transported or imported from specific place to specific place. In most cases, we need them to move from the extracellular fluid to the intracellular fluid. When you eat your food, the food molecules, the glucose, the fatty acids, and the amino acids, eventually get into the bloodstream, and we'll talk about how that happens later on. But they get into the bloodstream, but where these nutrient molecules really need to go is they need to go from the blood, which is an extracellular fluid, to the insides of cells, which is intracellular fluid. So they obviously need to pass through the cell membrane. Glucose, fatty acids, and the like need to be able to move through the cell membrane. Oxygen has an easy time doing so for reasons that we'll talk about soon. When cellular respiration occurs, we make waste products like carbon dioxide and urea, which need to be removed. They need to move in the opposite direction. We need to get them from the intracellular fluid out into the extracellular fluid so we can expel them out of the body. Signaling molecules like hormones, which we just talked about, have to be made inside of a particular endocrine gland, inside the cells of that gland, and then secreted, meaning it is exported from the intracellular fluid to the extracellular fluid. And then that hormone needs to travel through the bloodstream and then get to a particular cell in the body that it is signaling to. And depending on what kind of hormone it is, that hormone may need then to go inside of that cell. And then the last thing is that, just as kind of a housekeeping order of business, the concentrations of everyday things like water and different ions like sodium, potassium, calcium, chloride, and bicarbonate, they need to be maintained at a certain normal range for various homeostatic reasons. So to maintain all of these things is going to involve different kinds of membrane transport processes happening at very specific times, which we are going to get into. Okay, so you will recall that when we first introduced the cell membrane, we said that it is selectively permeable. It lets certain things pass through while restricting the passage of others. Well, how does it decide that? Which sorts of things get through and which ones get blocked? So thankfully, we actually have a set of rules that govern what gets in, what gets out, and under what circumstances. Now, the first clue to coming up with this set of rules is what we talked about in the first video, the actual chemical structure of the phospholipid bilayer. That is our first level of control over membrane transport. If a molecule is going to move straight through the membrane, we need to keep in mind that that molecule is going to have to interact with those hydrophobic tails in the core of the phospholipid bilayer. So if you're thinking about a molecule moving straight through the plasma membrane, it would do so by squeezing in through the space in between phospholipid heads. And as I was indicating, it's going to have to spend some time interacting with these hydrophobic tails. So that might get you kind of thinking about what kinds of molecules would have an easy time doing that. So only very specific solutes that meet very strict criteria are going to be able to do this. And by this, I mean pass directly through the membrane without any sort of assistance whatsoever. So this is exactly why the membrane is described as being selectively permeable. Some molecules, like the ones that I'm indicating here, can actually pass straight through without any sort of assistance. Other molecules are going to need some help. So let's go ahead and lay these rules down. The membrane is going to discriminate molecules on the basis of 3 different qualities. These are the qualities that the membrane is going to consider when deciding whether to let a molecule straight through or not. The first is kind of obvious. How big is the molecule? Molecular size. And this one is basically just a whole bunch of common sense. You probably don't even need me to tell you this. The larger the molecule is, the more difficulty it's going to have passing through the bilayer. Consider that for a molecule to get straight through, it has to pass in between. It has to essentially wedge itself in between 2 adjacent phospholipids. The bigger the molecule is, the more difficult that is going to be. So all other things being equal, a single atom, which is very, very small, would easily pass through, whereas a very big protein, which contains thousands and thousands of atoms, would have a very, very difficult time. So the first thing we're considering is how big is the molecule, a pretty simple thing to consider. Number 2 is polarity. So we're talking about polarity again, bringing that discussion back in from chapter 2. Now, this leads into what I was just talking about on the previous slide. If a molecule is going to pass straight through the membrane, it has to interact with those nonpolar tails that each phospholipid has. And those tails are nonpolar, and they are not going to like the idea of a polar molecule trying to come through. They will repel polar molecules. And because of this, the more polar a molecule is, the more difficulty it's going to have trying to squeeze in between the phospholipids. So it's one thing to say that a molecule is very small, but if that very small molecule is very polar, then the size kind of becomes a moot point at that point. So far, looking at molecular size and polarity, the ideal molecule to pass straight through the membrane without any assistance is one that is very small and one that is as non-polar as possible. Being a big molecule and being a polar molecule is not good for trying to get through the membrane all by yourself. And then the third one is actually fairly simple. Electrical charge. This one is very, 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 very black and white. Now, polarity and size are things that fall on a scale. You've got some molecules are bigger than others, and then some molecules are even bigger or even bigger. Polarity falls on a scale too. Electrical charge is pretty simple. You're either electrically charged or you are not. In fact, you can consider electrical charge as just being a very extreme example of polarity. Polar molecules have partial electrical charges, and it's those partial charges that prevent polar molecules from getting through the membrane. So if you're talking about something like sodium or potassium that have full electrical charges, that's going to be full stop. Anything that has a full electrical charge does not have a chance in hell of getting through the membrane all by itself. It's going to need some assistance in the form of carriers or channel proteins. So to sum it up here, if we were drawing up an ideal molecule that gets through the membrane all by itself as easily as possible, it would be very small, it would be totally nonpolar, and it would be electrically neutral. Now here's a picture that kind of illustrates what we were just talking about, what sorts of molecules get through and which ones do not. So you can see some of the important things here. A very, very, very big molecule like a protein or some other macromolecule, it doesn't matter about its polarity or its electrical charge, it's just too big. It can't squeeze in between the phospholipids, so it gets repelled. It's stuck on one side or the other. Small, non-charged, non-polar molecules can pass straight through without any assistance, and you can see they just squeeze in between the phospholipids. Molecules that have full electrical charges are totally repelled in the same way that polar molecules are. Now, the thing that I don't really like about this figure is what it is indicating for water. It's showing that water freely passes straight through the plasma membrane. But what's the problem with that? The problem is that water is a very polar molecule, and as such, this figure is quite a bit misleading. It gives the impression that in spite of water's polarity, it freely moves through the plasma membrane. Now, that's not really the case, and we will clear that little issue up in the next video when we start talking about osmosis. So, this is a very useful figure if you want to use it for study purposes, but don't take it literally that water moves through the membrane all by itself. It's going to be much closer to the charged molecules than this non-charged molecule right here. Okay, and then another important question to answer when we're talking about membrane transport processes is when and how and why do molecules move across the membrane? Do they just do it whenever they feel like it? Does it just happen randomly? How do molecules move across? Well, we know how they move across the membrane, but when and why do they move across the membrane? It turns out there is a specific force that dictates in which direction molecules move and how quickly they do it. The net direction in which molecules freely move depends on something called a concentration gradient, or a difference in concentration for that particular molecule on either side of the membrane. And this is why it was so important for us to get a good understanding of what concentration means in the previous chapter. We're talking about how many molecules are in a certain volume of fluid. So look at the picture here. We've got a plasma membrane that is separating an extracellular fluid from the intracellular fluid. Extracellular fluid on top, intracellular fluid on bottom. In blue here we have a just generic type of solute, a small, non-charged nonpolar molecule, the type of molecule that can move straight through the membrane without any assistance. And that's going to be easiest for us to work with conceptually here. Now, what you'll notice at the far left of this figure, when time first begins, so time is kind of going on as you go from left to right, at the very beginning of this little demonstration here, the concentration of this blue molecule is very high in the extracellular fluid and very, very, very low in the intracellular fluid. In fact, we could say the concentration is 0 down here because there are no molecules down here. So the concentration is very high on one side of the membrane and very low on the other side of the membrane. This is exactly what we mean by a concentration gradient. We're talking about a discrepancy or a difference in concentration for one particular molecule on one side of the membrane versus the other. So this concentration gradient is going to provide the force that is going to cause these molecules to move across the membrane. It's just the natural order of things, and you'll know what I'm talking about here. If you find yourself in a very, very, very crowded room, your natural inclination is going to be, I got to get the hell out of here. I've got to get some fresh air. I got to get some elbow room. You don't want to be cooped up with a whole bunch of other people. Who wants that? Nobody. So you want to get away from all of those people, and you want to kind of give yourself some breathing room. Well, molecules are kind of the same way. They don't really like to be all cooped up with each other if they have an alternative. And for these molecules up here, the alternative is that they're saying, hey, we're all crowded together. Well, why don't we space out a little bit? Why don't some of us go down here where there's a lot more extra room? So this is the natural force that is going to cause molecules to move across the membrane. They follow the concentration gradient. The concentration gradient is essentially pulling these molecules from the area of high concentration down towards the area of low concentration. This is what, and I'm going to say this several different ways throughout the semester, you can either say that these molecules are following their concentration gradient or they're going down their concentration gradient, kind of like you ride a bike downhill. Riding downhill is nice and easy. Molecules moving down their concentration gradient is nice and easy as well. So solutes are just naturally going to diffuse from areas of high concentration to areas of low concentration, provided they can move. Keep in mind here, we are specifically told this is a small, non-polar, non-charged molecule, so we know it can move. But what happens if we're talking about a solute that cannot move? Something for us to think about. Now, these molecules are going to move down their concentration gradient, but they're not going to do that for forever. So these molecules are going to keep moving until we get to the point where we establish an equal concentration of this particular molecule on both sides of the membrane. So you can see we've got an equal concentration in the extracellular fluid and an equal concentration in the intracellular fluid. At this point, we would say there is no longer a concentration gradient. These solutes will continue to move in both directions, but at equal rates, so the concentrations will not change. When we do this, we have a specific term for what we have accomplished. This is called establishing equilibrium. At equilibrium, you have an equal concentration of solute on both sides of the membrane. Solute, like I said, will continue to move at equal rates in both directions so that the concentration on each side does not change. Now, something that's important to consider is that even though concentration gradients and the diffusion of molecules across the membrane are done so that we try to establish equilibrium, but remember what homeostasis is all about. Things are always trying to change, but we try to resist those changes. So establishing equilibrium in most cases, not all cases, but most cases is actually a bad thing. So we have other cell transport processes to try to prevent this equilibrium from occurring, and we will talk about those as we go on. Okay, so essentially what we have just got done describing here with this figure is what is called simple diffusion. It is a type of diffusion that falls under the umbrella of passive transport. Passive transport is any membrane transport process that allows a molecule to follow or go down its concentration gradient. As long as a molecule is moving down its concentration gradient, it is passive transport. And because we are going downhill like this, there is no energy input required. We don't have to burn through ATP. We don't have to spend any energy to get any of this stuff done. It just happens naturally, just like riding your bike downhill. You don't have to pedal your bike when you go downhill. Gravity will just pull you down naturally. So passive transport includes two different types of transport within it. The one we just got done describing here is called simple diffusion. Simple diffusion involves molecules moving down their concentration gradient, and these molecules that we're describing with simple diffusion move through the membrane all by themselves without any assistance. So that little exercise that we went through a few minutes ago talking about how big is a molecule, how polar is it, and is it electrically charged, those are really the rules that establish whether a molecule moves by simple diffusion or not. So if the solute can pass through the membrane all by itself without any assistance whatsoever, like oxygen does, then that process is called simple diffusion. A good example of simple diffusion in action is gas exchange in the lungs. Another type of gas exchange process also occurs in the tissues, but we'll go ahead and focus on the pulmonary gas exchange in the lungs. Now, this is going to involve oxygen and carbon dioxide, which are both very small molecules and they're very nonpolar, so they are exactly the kinds of molecules that can move through the membrane by simple diffusion. They're moving in opposite directions here. So why are carbon dioxide and oxygen moving in opposite directions here? Well, let's go ahead and set the stage here so we have a better idea of what we're talking about. So when you breathe in, if you want to, go ahead and take a big deep breath in. When you breathe in, your lungs, which are made up of thousands, if not millions, of these little small sacs of tissue called alveoli, kind of like little inflatable balloons, when you take a deep breath in, you are filling these alveoli with air, which contains both oxygen and carbon dioxide. Well, your blood, which is running through capillaries that pass very close by to these alveoli, your blood also contains oxygen and carbon dioxide. So in determining as this blood moves by, and you can see we've got cell membranes that separate the air in the alveoli from the blood in the capillary. In determining which direction oxygen moves and which direction carbon dioxide moves, we have to consider their concentration gradients separately. The air that we breathe contains 21% oxygen. It doesn't sound like a lot, but it is a ton of oxygen. It is plenty of oxygen. The concentration of oxygen in the air here is much greater than the oxygen in the deoxygenated venous blood that is coming from the right side of the heart, from the right ventricle. So oxygen is low here, oxygen is high right here. So oxygen is going to move from an area of high concentration inside the alveoli, across the membranes of the alveoli and across the membranes of the cells that make up the capillary, and that oxygen is going to diffuse into the blood. That's why the blood that comes out and goes back to the left atrium of the heart is now oxygen rich, because we have taken oxygen from the air that we just breathed in. That oxygen diffused across the membranes that make up these barriers here, and the oxygen got into the blood. So now we have oxygen-enriched blood going to the left side of the heart, and we're all ready to pump that out to all of the needy tissues in the body. So don't forget here, an important aspect here is that oxygen moves through these membranes very, very easily by simple diffusion. It's small, it's nonpolar, it has no electrical charge. Carbon dioxide is very much the same way. It's small, it's electrically neutral, and it's nonpolar. But the difference here is that carbon dioxide's concentration gradient points in exactly the opposite direction. The venous blood coming from the right side of the heart actually has quite a lot of carbon dioxide in it, more carbon dioxide than what you find inside the alveoli. So what is going to be the case here, just as it was with oxygen, oxygen moved from high concentration to low concentration. Carbon dioxide is also going to move from high concentration to low concentration. But the problem here, well, not really a problem, just kind of something that we need to make sure we understand, is that the concentration gradients are flipped compared to one another. The concentration gradient for oxygen, high here, low here, pulled oxygen into the blood. The concentration gradient for carbon dioxide, high in the blood, low in the alveoli, is going to pull carbon dioxide out of the blood. So that's an important concept to make sure that you understand. You treat concentration gradients separately for every new molecule that you're talking about. The concentration gradient for oxygen is going to have no influence on what carbon dioxide does. The concentration gradient for glucose has nothing to do with the concentration gradient for sodium. So for each unique solute that can move through the membrane, you treat its concentration gradient separately from the concentration gradients of other molecules. So in this case here, for both oxygen and carbon dioxide, there are two different sets of membranes that are being crossed. The membranes of those cells that make up the alveoli, which are very kind of thin and flattened here, so those are membranes that you see right there, and then the membranes of the capillary endothelial cells that actually make up the little small tube that carries blood through the alveolar network here. So just make sure that you understand the concentration gradient aspect of this here, why oxygen goes one way and carbon dioxide goes the other. This specific gas exchange process is something that we'll revisit in a lot more depth when we get to respiratory physiology at the end of the semester. So don't worry about the rest of that. So recall that I said that there are two types of passive transport. Passive transport, again, all it means is that a molecule is moving down its concentration gradient. That's all it means. Passive transport, no energy required because the solute is moving down its concentration gradient. Simple diffusion, the first kind of passive transport, required that a molecule can move through the membrane all by itself. So the next kind, which is called facilitated diffusion, deals with molecules that want to move down their concentration gradient but cannot move through the membrane all by themselves. So these are the kinds of molecules that fail one of those tests that we were giving earlier. Glucose, for example, is a small molecule. It's not electrically charged, but it is very polar, so it can't move through the membrane all by itself. Sodium is extremely small, but it's also electrically charged, so it can't move through the membrane all by itself. So when we have concentration gradients for molecules like these that cannot move through the membrane all by themselves, the only way to get them through the membrane is by using facilitated diffusion, at least as long as we're talking about passive transport. So what will become necessary for getting these solutes across the membrane is either a channel or a carrier protein. We need those channel or carrier proteins to be in the membrane, and we need them to be open for business, just like we said earlier. So having a channel or a carrier protein that lets the solute of interest through the membrane, this will allow that particular solute to bypass the hydrophobic core. For something like glucose or sodium, the reason why it can't pass straight through the membrane is because the hydrophobic tails of the hydrophobic core will not let them. Don't forget, nonpolar, polar. But if we move through a channel or a carrier, we bypass that. These hydrophobic tails are going to have no idea what happened because they can't see through the outer part of that channel. So it's like we're sneaking glucose or sodium or whatever through the membrane without letting those hydrophobic tails know. It's kind of like when you used to sneak out of your room at night to get cookies out of the cookie jar, and your parents hopefully were none the wiser at that point. So this specific facilitated diffusion specifically applies for polar and electrically charged solutes, since we've already established that they cannot cross the membrane directly. This does not apply to very big molecules like proteins. Proteins are too big to move through another protein. the more you kind of think about it, the more that kind of makes sense. So proteins are going to have to use exocytosis and endocytosis, which is a part of your on your own work for this chapter. Okay, so what you might be wanting to think here is that for facilitated diffusion, you might be thinking, since we're having to provide assistance in the form of a channel or a carrier, that must mean that there is an energy requirement. There is no energy requirement. As long as we are still moving down the concentration gradient, there is no specific energy requirement. What we're going to eventually find out here is that the only reason why you would have to burn energy, ATP or otherwise, for a membrane transport process is if you're moving a solute up its concentration gradient from an area of low concentration to an area of high concentration. That goes against the natural order of things, so you have to kind of spend a little bit of money, you kind of have to grease the wheels a little bit to get that to happen. Okay, so what we're going to do here before we wrap up this video is just introduce the idea of osmosis. And just very quickly here, I think, yeah, so we're almost done here. So excuse me for having to jump ahead there. So before we kind of get into osmosis in the next video, which is in and of itself a very dense topic, let's just go ahead and introduce osmosis itself. So this brings us back full circle to what I was talking about with water. I wasn't really a big fan of that one particular figure that showed water moving through the membrane all by itself. There's a problem with that. Water is, of course, a very small molecule, so just on the basis of its size, it ought to be able to move through the membrane very easily. But it is also a very, very polar molecule, so it should be blocked from moving by simple diffusion in that regard. Well, the reality is that water can cross through the membrane by simple diffusion, but it happens extremely slowly. Very, very slowly because of its polarity. If it wasn't nearly as polar, it would move through much quicker. So the deal with this is that we can't just count on osmosis, the movement of water across the membrane. We can't just count on osmosis to occur by simple diffusion. It's going to be too slow to really play a role in physiological processes. So that's why osmosis needs to be sped up. And the way we speed up osmosis is by getting it to occur by facilitated diffusion. What do you need for facilitated diffusion? You need channel or carrier proteins, right? And it just so happens that there is a very specific water channel protein that is there to just let water molecules through the membrane if there is a reason for water to move. And these water channels are called aquaporins. Very easy name to remember. Aqua means water, and porin means we're making a pore in the membrane for water to move through. So this figure is a lot more accurate, and it a lot more accurately describes how water moves through the membrane. Now, you can see other things move by simple diffusion, like oxygen and carbon dioxide we already mentioned. Steroid hormones are very small and hydrophobic, so they can move through as well. But because water is very polar, it's not going to be able to move through the membrane very easily unless there is an open aquaporin channel there. And I say an open aquaporin channel, but aquaporin channels are always open, so you don't really have to worry about them ever being closed. All right, that's going to do it for this video. We will start next time by talking more about osmosis and some of the consequences behind what happens when water moves from one side of the membrane to another, and how oftentimes that's going to be a bad thing, and we want to, in most cases, prevent that. All right, thank you for your attention, and I will see you in the next video.